Cytochrome P450 is an enzyme that in humans is encoded by the CYP2D6 gene. CYP2D6 is primarily expressed in the liver. CYP2D6, a member of the cytochrome P450 mixed-function oxidase system, is one of the most important enzymes involved in the metabolism of xenobiotics in the body. In particular, CYP2D6 is responsible for the metabolism and elimination of approximately 25% of clinically used drugs. This enzyme also metabolizes several endogenous substances such as hydroxytryptamines and neurosteroids. There is considerable variation in the efficiency and amount of CYP2D6 enzyme produced between individuals. Hence, for drugs that are metabolized by CYP2D6, certain individuals will eliminate these drugs quickly while others slowly. If a drug is metabolized too quickly, it may decrease the drug's efficacy, while if the drug is metabolized too slowly, toxicity may result. Hence, the dose of the drug may have to be adjusted to take into account of the speed at which it is metabolized by CYP2D6. In addition, other drugs may function as inhibitors of CYP2D6 activity or inducers of CYP2D6 enzyme expression that will lead to decreased or increased CYP2D6 activity respectively. If such a drug is taken at the same time as second drug who is a CYP2D6 substrate, the first drug may affect the elimination rate of the second through what is known as a drug-drug interaction. Gene. The gene is located near to cytochrome P450 organs on chromosome 22q13.1. Alternatively, spliced transcript variants encoding different isoforms have been found for this gene. Genotype-phenotype variability. CYP2D6 showed the largest phenotypical variability among the CYPs, largely due to genetic polymorphism. The genotype accounts for normal, reduced, and non-existent CYP2D6 function in subjects. Pharmacogenomic tests are now available to identify patients with variations in the CYP2D6 allele and have been shown to have widespread use in clinical practice. The CYP2D6 function in any particular subject may be described as one of the following: poor metabolizer little or no CYP2D6 function, intermediate metabolizers metabolize drugs at a rate somewhere between the poor and extensive metabolizers, extensive metabolizer normal CYP2D6 function, ultra-rapid metabolizer multiple copies of the CYP2D6 gene are expressed, and therefore greater than normal CYP2D6 function. A patient's CYP2D6 phenotype is often clinically determined via the administration of dabrissocine and subsequent plasma concentration assay of the dabrissocine metabolite. The type of CYP2D6 function of an individual may influence the person's response to different doses of drugs that CYP2D6 metabolizes. The nature of the effect on the drug response depends not only on the type of CYP2D6 function, but also on the extent to which processing of the drug by CYP2D6 results in a chemical that has an effect that is similar, stronger, or weaker than the original drug, or no effect at all. For example, if CYP2D6 converts a drug that has a strong effect into a substance that has a weaker effect, then poor metabolizers will have an exaggerated response to the drug and stronger side effects. Conversely, if CYP2D6 converts a different drug into a substance that has a greater effect than its parent chemical, then ultra-rapid metabolizers will have an exaggerated response to the drug and stronger side effects. Genetic basis of variability. The genetic basis for extensive and poor metabolizer variability is the CYP2D6 allele, located on chromosome 22. Subjects possessing certain allelic variants will show normal, decreased, or no CYP2D6 function, depending on the allele. Pharmacogenomic tests are now available to identify patients with variations in the CYP2D6 allele and have been shown to have widespread use in clinical practice. Ethnic factors in variability. Ethnicity is a factor in the occurrence of CYP2D6 variability. The prevalence of CYP2D6 poor metabolizers is approximately 6–10% in white populations, but is lower in most other ethnic groups such as Asians. In African Americans, the frequency of poor metabolizers is greater than for whites. The occurrence of CYP2D6 ultra-rapid metabolizers appears to be greater among Middle Eastern and North African populations. This variability is accounted for by the differences in the prevalence of various CYP2D6 alleles among the population. Approximately 10% of whites are intermediate metabolizers, due to decreased CYP2D6 function, because they appear to have the non-functional CYP2D6 4 allele, while approximately 50% of Asians possess the decreased functioning CYP2D6 10 allele. Ligands, following is a table of selected substrates, inducers and inhibitors of CYP2D6. Where classes of agents are listed, there may be exceptions within the class. Inhibitors of CYP2D6 can be classified by their potency, such as strong inhibitor being one that causes at least a 5-fold increase in the plasma AUC values, or more than 80% decrease in clearance. Moderate inhibitor being one that causes at least a 2-fold increase in the plasma AUC values, or 50 to 80% decrease in clearance. Weak inhibitor being one that causes at least a 1.25-fold but less than 2-fold increase in the plasma AUC values, or 20 to 50% decrease in clearance. References. Further reading. External links, Flockhart Lab CYP2D6 Substrates page at IUPUI, PHARMGKB, annotated PGX gene information for CYP2D6.